Hello and welcome to New York City Atheists Live on Tape, brought to you by New York City Atheists Incorporated. I'm your host, Dennis Horvitz, the David Letterman of the Hopelessly Damned. Without further ado, please welcome Richard L. BOROWSKY, Professor of Biology at NYU. We're going to find out why flying fish are lying.
Oh, I'm not going to talk about any of that.
We have evolved beyond it.
So, in the last couple of minutes, I've decided that, actually half an hour or so, I've decided that this is really a tough room. It's more shaped like a bolso, about the size of the other one. This comes from Brazil. So I have good colleagues and good relationships with people in Brazil and Thailand and Mexico, and these are the places where I do my work. This shows another species, also Brazilian, not quite as light in color, filmed in the cave. The water it's dealing with isn't moving quite as quickly as the water that Cryptotora dealt with, but these guys are quite capable of exploiting the environment that they're found in and where they live, and have a characteristic look that all of us know denotes that they're cave fish. And then the final few examples. This is a karst formation in central Thailand. It's overlain by harder rock, which is how it's survived, whereas everything around it has been eroded away. And there are caves in this formation, and we've collected different species of cave fish there. Some of them are completely eyeless, or at least externally eyeless. Some of them of the same species have small eye spots, non-functional, but all these fish I've shown you may have a little bit of yellow coloration or pink coloration, but they tend to lack melanin, and they certainly lack a visual system. And that, of course, comes about from the environment in which we live, but there are questions about exactly what were the forces driving the evolution of this eyeless condition. And just to make the point that there are certain other interesting aspects of these caves. Lots of the caves in Thailand and some in Mexico have bad air, so you're breathing 4 or 5% carbon dioxide and existing on 17% oxygen, which is actually not so hard to do. It's a little bit painful, but if you're not exercising hard, it's okay. If, however, you rappel into it, you really find it very, very difficult to climb out of it. And this is Jerry Campbell, who's in charge of caving safety. I say he's going the extra mile right before we enter one of these caves. Was that a grr I heard? I forgot for a moment who I was talking to. Well, that's Jerry's thing. Cave fish exhibit this convergence in loss of eyes, in loss of pigmentation, and in other characters. They've changed, it's not as obvious, but they've changed their metabolism. They're far more efficient, because if there's not much food around, you really better be able to make the maximum use of what you can get. And I guess this will be the final final slide on convergence. So the bread and butter species, these Mexican cave fish, come from this area in Mexico and are found in, they're not all shown here, but 29 different caves in this area over 140 kilometers from here, the northernmost cave, to the southernmost cave. They actually have derived from at least 2 separate species of surface fish that have been in the area. There was one set of surface fish that inhabited the streams around these caves and gave rise to populations over here. Then they went locally extinct. Another one came in and gave rise to populations here and there. So independent invasions. This is an old slide. I say 4 separate origins. We now know it's 5. Independent invasions, independent stocks, and yet they all converged on very much the same phenotype, the same look. One of the convergences, remarkably enough, is for sleep. So this is a record of activity of animals, of 4 different groups of animals, over a 2-day period. So the daylight, 12 hours, nighttime, 12 hours, the second day, 12 hours, second night, 12 hours. And what's plotted here is the amount of activity, the amount of inactivity, sleep, during 10-minute periods throughout this time period. And you can see that in black, surface fish sleep at night. Most of those 10 minutes at night are spent not moving, sleeping. Same for the second night here. They sleep much less during the day, although they seem to take the Mexican fish. They seem to take a siesta around midday. And in colors, red, whatever this is, gold and blue, we have the same record, same type of record for 3 different species, 3 independent populations of cavefish. They all have very much reduced amount of sleep. Here is a bar graph. Surface fish sleep 800 minutes a day, and the 3 independently derived populations of cavefish sleep 200 or 100 minutes a day. We don't really know why this is the case. We think that if you're in an environment where there's not much food and you can't predict when the food will come by, you better be awake when it does come by in order to get it. But that's just a hypothesis. We haven't tested that yet. So it's not only eyes and pigmentation, it's metabolism, it's behavior, it's lots of things that have changed. Am I speaking too long? No. Okay. So as was shown in the previous graph, for sleep, we're looking at 3 different independently derived cave populations of the same species where you have convergence. And the convergence is shown here. Here's a fish from Molino Cave, most northernmost of these Mexican caves that have cavefish. And then here's one from the Patron population. These guys are albino, these guys are albino. These guys are completely blind. These guys are completely blind. They both have very small residual eyes, or I'm sorry, I'm searching for a term, but that'll do. Sunken deep in the head, and they certainly look very much alike. I can actually tell the difference between them when they're swimming around in the tank, but you probably would not be able to. So this convergence that I speak about is really very well exhibited by this group. And I think this is the point I made in the introduction. I meant to make it here. Convergence is to evolutionary biology as replication is to experimental science. 100 different species of cavefish is 100 replicates of the same experiment. One species with 29 populations, that's 29 replicates of the same experiment, where because they all came from the same species of surface fish, you have a better idea of what their genetics is. So, Darwin. Darwin, when he wrote The Origin, knew about cavefish. He also knew about cave salamanders from Slovenia, but in the United States, there's a group of cavefish that had just been discovered, and obviously these were of great interest to him. He knew, as well as everybody did, everybody believed in evolution or change through time, he knew that they had descended from surface fish. And so he knew the polarity of the change, and what he's saying here, much more eloquently than I could say it, is if you poke my eyes out and put me in a cave, if you put me in a cave, I can't see. If you poke my eyes out, I can't fail to see any more efficiently than I failed to see when I had my eyes. It's dark in a cave. So he's saying, I can't see how eyes are injurious to animals living in darkness, and he knew nothing about genetics. He had actually been mailed a copy of the journal that had Mendel's paper in it, but he never actually read that paper. And so he simply had to attribute the loss wholly to disuse. We now know, of course, much more about genetics, or we know about genetics, and we know from Darwin about natural selection, and we know other means, not driven by natural selection, by which a character can, that isn't needed, can degenerate. And so there's some residual debate as to whether eyes are selected against in the dark or not having a function. Natural selection will not be policing and screening new mutations that come along. So new mutations might come along, not be selected for, but just by chance increase in frequency in the population, and structure tends to run downhill if it's not tended. The metaphor I used before was the metaphor of the untended house. If I move away from my house for 20 years and nobody's watching it, I mean, I know for sure when I come back there'll be window panes broken, there'll be leaks in the pipes, there'll be squirrels in the roof, there'll be there are squirrels in the roof. But what I don't know is where these window panes will be. I don't know which tiles or shingles will be pulled up by the squirrels and so forth and so on. So if natural selection is not policing a phenotype, keeping it up to snuff, keeping it up to par because it's needed, in this case because it's not needed, then things can go downhill. That's called genetic drift as opposed to natural selection. So major questions, I'm not sure I'll cover them directly, but if it is selection, how? Referencing Darwin's dilemma, I don't really understand it, he said. Mutation, is it random mutation, is it drift? I've explained it all. And one of the things that we're interested in as well is how many genes have been changed between being an eyed surface animal and a cavefish. Is it one gene that's knocked out, or 2 genes, or 10 genes? Before we started this research, nobody actually knew. Now we know it's probably on the order of 10, 12 genes per population. So I haven't any memory of why I put this in here. I assume that I'm going to show you fish from Tinaja, Pachon, and Molino, or something like that. Oh, yeah. So here's our surface fish. We know the polarity of change. Here's the Molino cavefish, which is an albino, as I mentioned. And here's one from a different cave, Tinaja cave. You can see a little bit of dark pigment in the mid-lateral line of that fish. These are actually not albinos, but they have greatly reduced numbers of melanophores, these pigment cells that are found in greater number in these guys. And when I hybridize them, what I obtain is a fish that in its pigmentation looks very much like the surface fish, maybe even darker than the surface fish, and very different than either of its parents. This is a phenomenon that's called complementation, and let me illustrate it with an example from human biology. Hearing disabled people tend to hang out with one another. I mean, they speak the same language. I can't do that, but they can understand one another with sign language. They tend to marry, as in all marriages, they tend to have children, and many times their children are also deaf. They're also hearing disabled. Sometimes, however, the child is able to hear. And that raised the question, is this guy the father of the child, or is somebody else the father of the child? And most of the time, he actually is the father of the child, 9 out of 10 times. And what it is she's deaf because gene A is bad. But gene B, which is also involved in hearing, is good. He's deaf because gene B is bad, but gene A is good. So he contributes in the sperm one good copy of A and a bad B. She contributes one good copy of B and a bad A. And the good copies of A and B are sufficient to make a hearing enabled child. And so that's complementation. The good qualities of one complement the bad of the other, and vice versa. And so here I have this diagrammatically for pigmentation. One of the fish, let's say Molino, has a good copy of A, but is homozygous. One copy from its mother, one copy from its father. Of B is little b, but it needs big B for dark pigmentation. It doesn't have it. Here, the fish from Tinaja is light in pigmentation. It has little a, and that's the deficiency. It needs big A, but it has big B. He contributes big A and little b, she contributes little a and big B, and the offspring is dark because it's complemented. That was a much simplified diagram. We now know that it's about a dozen factors in each population. But these dozen don't, I wish I had 3 hands, these dozen don't overlap with these dozen, and therefore you have this complementation. The complementation is also seen in vision, and so here's a 2-minute video illustrating this. Unfortunately, I can't play the soundtrack, but I can narrate it for you.
The Mexican tetra has 2 forms.
The Mexican tetra has 2 forms.
Cavefish that lack external eyes and are blind. Purebred cavefish are always blind.
I feel like I'm dowsing. I'm looking for the for the microphone. But the purebred cavefish always are blind. When you hybridize them, however, you can get fish that can see. So what we did is we tested them in this little apparatus with this cup with white and black barrel staves. You put the embryo, because we worked on embryo on fry, you put the fry in the bottom, you whirl the cup. If the animal can see, its eyes follow the stripes and snap back, follow and snap back. So this is a surface fish, very small fish. Big eyes, you can see them flicking, and in this bubble and in the shadow on the bottom you can see the movement flicks. This is a cavefish with small eyes, and you can see the bubble is, reflected in the bubble is turning. The eyes aren't moving at all. It's really blind. This is another cavefish. You can see the movement over there, but the eyes aren't moving. So he's blind. And then I'm going to show a hybrid between those 2 cavefish forms, and you'll notice that the eye spots are much larger. So eye spots are larger, and you can see them flicking as the surface fish eyes flick. So this is one generation of hybridization, one cave and another cave. Just as with pigmentation, these guys are blind because of changes in a certain set of genes. Those guys are blind because of changes in a different set of genes. In the hybrid, you get this complementation.
These experiments show that fish can be restored cavefish if you make the right genetic crosses. So learn more about this exciting.
So it's actually the same diagram that I showed before to explain restoration of pigment, in that it was picked up by Richard Dawkins. He really liked that story. And it was also picked up by a website called Creation Safaris. The creationists really like evolution in cavefish. Their argument is that only God can make something as complex as an eye, but random mutation can break down something that God has made and destroy it. So they're perfectly happy with the idea of regressive evolution. And in fact, their synopsis of my paper, I thought, was the best written and clearest of all the synopses that I read. It was really quite remarkable. And I know who I'm talking to, and I feel the same way myself, but it really quite amazed me. So, 3 conclusions, and I can shut up and take questions, or go. First, convergent populations or species can profitably be analyzed as replicates of evolutionary experiments. And I guess I haven't really shown that. It would be what I would talk about next if we had time for it, but maybe we don't. In cavefish, okay, this is the part that I haven't really explained to you. Direct selection is implicated in eye regression. And the third is that people can read into any story whatever they feel like. If you'll give me 3 more minutes, I will summarize what I would have shown you on those other slides.
Can you talk about what you mean by direct selection?
Okay, if you give me 3.5 minutes, I'll show you. So, if at any genetic locus you hybridize a cave fish with a surface fish, in the F1 hybrid you have heterozygote. It's cave surface. It's gotten one gene from one parent, another copy of the gene from another parent, and the other copies, we use the term alleles. The alleles are different. So gene has actually two meanings. One is a place on the chromosome, a genetic locus. So we might talk about the eye color gene. It's here in the genome. The hair color gene, it's there in the genome. But at that eye color locus, you can have a gene for blue eyes or brown eyes or whatever. And that second meaning of the word gene, we use allele for, different alleles. Then if you breed an F1 with an F1, you get a 1 to 2 to 1 ratio of homozygote, heterozygote, homozygote. If you test enough genetic loci throughout the genome, you can use the information from that experiment to put together a picture of the genome. So here's one of the linkage groups or chromosomes in that genome, and here's a gene called OCA2, which is the gene for albinism we've been able to show in those fish. And here's something called 218E, which is just an arbitrary designation. All of those numbers are arbitrary designations for loci that we can demonstrate. And the whole map would be far more extensive. It would have 25 of these linkage groups. We have 23. They have 25. And given the map like this, we can ask, does it matter at 105B whether an individual is cave-cave, cave-surface, or surface-surface? So is there a difference in eye size between those three? And most of the time, the answer is no. So when we look at gene 205E, the CC individuals, cave-cave at that locus, are medium, have medium-sized eyes. The heterozygotes have medium-sized eyes. The other homozygotes have medium-sized eyes. And what that means is that 205E has nothing to do with controlling the eye size differences. And it's not near anything in the genome that has anything to do with controlling those eye size differences. But when we look at 218E, these individuals in this class tend to have smaller eyes than those in the heterozygous class, which tend to have smaller eyes than those in the homozygous surface class. What that means is that near 218E, there's a gene that actually is controlling the development or the maintenance of the eyes. So something that's responsible. And so we use this sort of analysis to find about a dozen such places in the genome that really were important for eye size. And here's the answer to your question. When we did this analysis for eye size, every one of those places we found had the same polarity, in the sense that the surface-surface class, each one of these lines it denotes the trajectory for an individual gene. The surface-surface class was always bigger than the cave-cave class. But when we did the same analysis for number of melanophores, we found that most of them were in, what we call the right direction, but some of them had exactly the opposite polarity. And the difference between this pattern and that pattern is statistically significant, and we replicated it, the importance of replication, and replicated it in analysis of another cave population, so we're pretty sure that fate didn't play a trick on us. And the interpretation of this consistency is that natural selection is doing the job. Every time one of those mutations comes along, it increases the fitness of its holder, and it increases in frequency in the population. Whereas here, we view it as a case of genetic drift. If you don't have a melanophore cell there, you're going to have some other cell there. It really doesn't matter what cell you have there in your skin. If you're in the dark, you're not making melanin anyway, so it doesn't matter whether you have a melanophore cell there. There are several predictions that come from this. One of them is, since natural selection changes gene frequencies very quickly compared to genetic drift, we would predict that eye size decreases more rapidly in a cave population once it gets into a cave than the number of melanophores. We've looked at 6 other species of cave fish, and this prediction is borne out. The second part of the hypothesis is reason. Why should eyes be selected against in the dark? The brain is the most active tissue in your body, and the retina has a higher metabolic rate than any other part of the brain. It's an extremely expensive tissue. In us, 10% of our rods and cones are shed every day, so we renew the photoreceptor layer 36 times a year. Now, if you put me on limited rations and took out my eyes, the savings in energy probably wouldn't amount to very much, because I'm a mammal, I have a high basal metabolic rate. But fish, when they're not moving, have almost a 0 metabolic rate, and I wouldn't be surprised if the retina uses up maybe 1% or 2% of its daily energy budget. Testable hypothesis. I have a colleague in Scandinavia who's now doing those experiments on oxygen uptake of isolated retinas. So does that answer your question? And so I've spoken too long. I thank you for your patience, and I'll take any questions you have.